All right, welcome back to This Week in Biochar.
Always a pleasure.
Now, as always, we're diving into the hottest topics. The ones you, the Biochar Today readers, are buzzing about.
Absolutely. Staying on the cutting edge.
Because, let's be honest, there's a ton of research out there. It can be overwhelming, but that's where we come in.
Right, cutting through the noise.
Exactly.
Yeah.
This week, we've got two particularly fascinating studies that really caught your eye. One's all about how biochar is impacting maize, and not just the yield, but the actual quality of the kernels.
Yeah, some really promising findings there.
And the second dives into how biochar, made from different materials, can rapidly hold on to ammonium, that essential plant nutrient.
A crucial aspect of nitrogen management.
So let's jump right in. The first study comes to us from Science of the Total Environment. It's a field trial out of Italy, and the researchers weren't just looking at biochar alone. They were exploring its synergy with other players in the soil.
Right, they wanted to see how biochar interacts with what's already there, in this case, beneficial microbes.
Exactly. And they weren't just adding any old microbes. They used what they called syncoms, synthetic microbial consortia, alongside some familiar friends, our buscular mycorrhizal fungi, or AMF for short.
So essentially, creating a kind of dream team in the soil, working together with the biochar.
It sounds like they were aiming for a supercharged soil environment. Like, how can we make things really thrive?
That's a good way to put it. The experiment itself was pretty comprehensive. They ran it over 2 growing seasons, 2020 and 2021 in Parma, Italy. They used a standard commercial maize variety, and the soil type was a silty clay loam.
Okay, so pretty typical conditions for maize production.
Right. But they also did something interesting with the nitrogen fertilizer. One year they used the full recommended amount, and the next year they cut it in half.
Interesting. So they were testing how this biochar microbe combo would perform under different nutrient conditions.
Precisely. They wanted to see if it could help compensate for reduced fertilizer inputs.
Makes sense. Now, the big question is, what kind of impact did this have on the maize itself? That's what caught the eye of Biochar Today readers.
Well, one of the most striking findings is the synergy. The biochar, the syncoms, and the AMF, they all seem to work together to create a really favorable environment for the soil microbiome.
Okay, so they were boosting the overall health of the soil.
Exactly. And this, in turn, had a positive impact on the maize growth and even the composition of the kernels themselves.
Fascinating. So they were getting more than just bigger plants.
Right. It seemed to be impacting the quality of the crop as well. And importantly, they found that introducing these syncoms didn't have a negative impact on the existing microbial community in the soil.
So that's great news.
Mm.
Because you don't want to create one problem while solving another, right?
Absolutely. It's all about maintaining that ecological balance.
So the native microbes were happy. But let's get back to the maize. How did it respond to this soil dream team?
Well, they measured a range of factors. The plants treated with both the syncoms and the biochar, they showed higher levels of chlorophyll.
Okay, and that's a good thing, right? Chlorophyll is essential for photosynthesis.
Exactly. So it's a sign of healthy, vigorous plants. They also measured heavier ears of corn and heavier individual kernels, which translate directly to a better harvest.
Ah, so more maize. That always gets attention. But it sounds like it wasn't just about quantity.
No, the quality of the kernels was also impacted, and this is where it gets really interesting. They found that the combination of biochar and syncoms actually changed the metabolome of the maize kernels.
Metabolome. Remind me what that means again.
Sure, it refers to the complete set of small molecules or metabolites within a biological sample. In this case, they're talking about the compounds found inside the maize kernels.
Gotcha. So what kind of changes did they see in the metabolome?
They found an increase in compounds that are known for their health-promoting properties, particularly those with antioxidant and defense capabilities.
So they weren't just getting more maize, they were getting potentially more nutritious and resilient maize.
Exactly. That's one of the really promising aspects of this study. They did a very thorough job in terms of the measurements. They looked at chlorophyll content, leaf transpiration rate, which is a measure of how much water the leaves are losing, the height of the plants, the number of ears they produce.
Wow, they really covered all the bases.
Yeah, all the factors that contribute to the final yield. They even measured things like the weight of 1,000 grains or how much moisture the grains contained.
No stone unturned.
Right. And importantly, they also checked for aflatoxins, which are harmful compounds that can sometimes contaminate maize.
And I'm assuming they didn't find any?
Thankfully, no. The levels were below their detection limit in all the samples.
That's good news. So they had a comprehensive view of the maize plants themselves, but they also looked at the microbes living around the roots.
Yes, they analyzed what's called the rhizospheric microbiota, which is essentially the community of microorganisms living in the soil right around the plant roots.
And why is that important?
Well, that's where a lot of the action happens in terms of nutrient exchange between the plants and the soil.
Okay, makes sense.
What they found was that the existing microbial community was pretty resilient to the treatments. They only saw minor shifts in a few specific combinations in the second year of the trial.
So basically, this biofertilizer approach didn't disrupt the established soil ecosystem.
Exactly. It seems to be working in harmony with the existing microbial community. Now, let's dive a bit deeper into those metabolite changes in the kernels. You mentioned they found a lot of differences. Can you break down some of those key compounds for us?
Absolutely. In the first year, particularly in treatments that included certain syncoms, they observed higher levels of phenolic acids, phenylpropanoids, and benzoxazinoids. These are all compounds often associated with plant defense mechanisms and also have antioxidant effects, which are potentially beneficial for us when we eat them.
Interesting. So multiple benefits there.
Yeah. Then, in the second year, they saw increases in amino acids, other beneficial acids, and again, those benzoxazinoids. But interestingly, they noted a decrease in some of those phenylpropanoids in certain treatments. And across both years, they consistently saw a reduction in a compound called azelaic acid.
Okay, so some consistent trends, but also some variations depending on the specific syncoms used.
Right. Now, you mentioned some complex-sounding ones like rutin and HMBOA4-GLC. What about those?
Those are quite interesting. Rutin, known for its antioxidant and anti-inflammatory properties, and HMBOA4-GLC, a type of benzoxazinoid that acts as a defense compound in plants. Both of these increased in the maize kernels that were treated with syncoms.
Okay, so even more evidence of enhanced defense capabilities.
Yes, and they also looked at the non-polar metabolites, which include things like fats and oils within the kernels. They found that the combination of biochar, one particular syncom called MCB, and AMF led to the highest levels of these compounds in the first year. Then, in the second year, they saw an increase in linolenic acid, an omega-3 fatty acid, in most of the treatments.
Wow, so they really delved into the intricacies of the kernel composition. Were there any consistent changes they observed in the syncoms-treated maize across both years?
Yes, some key biomarkers emerged. They consistently found increased levels of D-pipeacolic acid, which plays a role in plant immunity, and specific classes of lipids, including trichoproin, a type of triglyceride. They also saw a decrease in certain other fatty acids.
Okay, so take it all together. What's the key message here for Biochar Today readers?
The main takeaway is that using syncoms, along with biochar and AMF, appears to be a safe and effective way to not only improve the yield of maize, but also boost its nutritional quality by increasing beneficial metabolites. And all of this without harming the existing microbial diversity in the soil.
So potentially a win-win for both productivity and sustainability.
Right, but it's important to note that the researchers themselves acknowledge we need more research to fully understand the economic viability and the broader environmental impacts in different real-world farming scenarios.
Of course, more research is always needed, but this is certainly exciting preliminary data. Now let's move on to that second study that caught the attention of Biochar Today readers. This one is about how quickly different types of biochar can grab onto ammonium. And it comes from the journal Sustainability.
Yes, this one is all about nutrient retention, specifically how well biochar can capture and hold on to ammonium, a vital form of nitrogen that plants need. They looked at biochars made from two very different feedstocks, coffee husk and chicken manure, and they produced these biochars at 3 different temperatures, 300, 400, and 900 degrees Celsius.
Okay, so they're looking at how both the source material and the production process affect the biochar's ability to capture ammonium. And I believe this study was done in Brazil, is that right?
You're right, and the choice of feedstocks is significant. Brazil has huge poultry and coffee industries, which generate a lot of chicken manure and coffee husk, making them potentially very sustainable sources for biochar production.
So it's about using what's readily available and turning it into something valuable.
Exactly. Now, in terms of the methodology, they first dried the raw materials and then pyrolyzed them, which means heating them in a furnace at those specific temperatures for an hour in the absence of oxygen. After cooling, they characterized the biochars, measuring things like their carbon content, pH, cation exchange capacity, or CEC, which is a measure of their ability to hold onto nutrients, and their physical structure, including surface area and pore size. They used a variety of techniques, including spectroscopy and microscopy, to get a detailed picture of these properties.
So like a thorough analysis.
Yeah.
And then how did they actually test the biochar's ability to capture ammonium?
They passed a solution with a known concentration of ammonium through the different biochar samples. But first, they washed the biochar with water to remove any loosely bound substances. They then analyzed the water that had passed through, measuring its pH, electrical conductivity, the amount of dissolved carbon, and the concentrations of ammonium, nitrate, and various other elements. They even used a computer model to predict if any solid compounds might have formed during the process.
Okay, so a comprehensive analysis of both the biochar and the water that passed through.
Yes. And they also analyzed the biochar itself after it had been exposed to the ammonium solution, using spectroscopy to identify the specific chemical groups involved in binding ammonium and measuring the total nitrogen content to see how much ammonium had been retained.
Okay, so a lot of data to analyze. What were some of the key findings regarding the properties of the biochars themselves? How did the feedstock and the pyrolysis temperature affect those properties?
Well, as expected, higher pyrolysis temperatures generally led to a higher percentage of carbon in the coffee husk biochar, but a lower percentage in the chicken manure biochar. The pH and electrical conductivity, both measures of the biochar's chemical environment, increased with temperature for both feedstocks. Interestingly, the coffee husk biochar had a higher cation exchange capacity, meaning it was better at holding onto nutrients at the lower temperatures of 300 and 400 degrees Celsius. However, this trend reversed at 900 degrees, where the chicken manure biochar showed a higher CEC. The chicken manure biochar always had more ash content, which reflects its mineral content, and this increased with temperature for both feedstocks. Now, for the physical structure, the coffee husk biochar had a larger surface area and more pore space, and these properties became even more pronounced at higher pyrolysis temperatures. However, the size of the pores tended to decrease at higher temperatures. They confirmed this porous structure using microscopy, especially for the coffee husk biochar produced at 900 degrees Celsius.
So a lot of interesting variations depending on the source material and how it was processed. But ultimately, the big question is, how well did these different biochars actually hold on to the ammonium?
Right. And that's where the different mechanisms come into play. The chicken manure biochar, produced at the lowest temperature, 300 degrees Celsius, started with the highest total nitrogen content. However, after they passed the ammonium solution through, it was the coffee husk biochar produced at the highest temperature, 900 degrees Celsius, that showed the greatest retention of the added ammonium. And they attributed this to its highly porous structure, essentially trapping the ammonium ions within those tiny pores.
So in that case, it was more about physical trapping than chemical binding.
Exactly. But they also found that the coffee husk biochar produced at 300 degrees Celsius, while not as porous, also showed good ammonium retention, likely due to electrostatic interactions with specific chemical groups on its surface.
Okay, so different mechanisms at work.
Yeah.
What about the chicken manure biochar? Did any of those stand out?
Yes, the chicken manure biochar produced at 400 degrees Celsius was interesting. It retained some of the nitrogen through the formation of a mineral called struvite, which contains ammonium, magnesium, and phosphate. They found evidence for this in the water that passed through, which showed high levels of magnesium and phosphorus at a pH suitable for struvite formation.
So in that case, the biochar wasn't just capturing ammonium directly, it was facilitating the formation of a new mineral that contained ammonium.
Precisely. And that highlights the complex interactions that can occur in these systems. They concluded that for the coffee husk biochar, higher temperatures, while reducing some of the chemical groups on the surface that can bind nutrients, actually created a more porous structure, which favored physical trapping. For the chicken manure biochar, the high mineral content at higher temperatures seemed to limit the development of that porous structure.
Okay, so it's not a one-size-fits-all solution. Different biochars, depending on their source and production, have different strengths when it comes to capturing ammonium. What's the key takeaway for biochar today readers from this study?
This research really emphasizes that biochar is not a single, uniform material. Its ability to capture and retain nutrients, in this case ammonium, varies depending on what it's made from and how it's produced. And these differences relate to the underlying mechanisms, electrostatic interactions, physical trapping, or even the formation of new mineral compounds. This understanding is crucial for optimizing biochar applications in different agricultural settings.
So tailoring the biochar to the specific needs of the soil and the crop.
Exactly. It's about matching the right tool to the right job.
So to wrap up our look at the highlights from biochar today, We've seen some really promising developments in the world of biochar research.
Absolutely. Both of these studies provide valuable insights.
The maize study shows that biochar, when combined with beneficial microbes, can not only improve yields but also enhance the nutritional quality of the crop.
And the ammonium retention study highlights the versatility of biochar in nutrient management, demonstrating how its properties can be tailored to optimize nutrient capture and retention.
It really makes you think about the potential of biochar in creating more sustainable and efficient agricultural systems. And it raises some intriguing questions. Knowing that we can fine-tune biochar properties based on the feedstock and production method, how can we best match those properties to the specific needs of different soil types and crops to maximize both productivity and environmental benefit?
That's a great question, and one that researchers are actively exploring.
It's definitely something to keep an eye on, and a great reason to stay tuned to Biochar today for the latest updates. Thanks for joining us on this week's deep dive into the world of biochar.
Always a pleasure.
See you next time.